The delivery of antigen from a site of infection to downstream lymphoid tissue and its subsequent presentation to naive T cells is actively aided by the innate immune response to infection. This is rapidly triggered at the site of infection by non-clonotypic receptors that recognize molecular patterns that are associated with pathogens but not host cells. One of the induced responses of innate immunity is an inflammatory reaction that increases the entry of plasma into the infected tissues and the consequent drainage of tissue fluids into the lymph. Another is the induced maturation of tissue dendritic cells that have been taking up particulate and soluble antigens at the site of infection. These cells are activated through receptors that signal the presence of pathogen components bound by dendritic cell receptors or by cytokines produced during the inflammatory response. The dendritic cells respond by migrating to the lymph node and expressing the co-stimulatory molecules that are required in addition to antigen for the activation of naive T cells. Macrophages, which are phagocytic cells found in the tissues and scattered throughout lymphoid tissues, and B cells, which bind pathogen components, may be similarly induced through nonspecific receptors to express co-stimulatory molecules and act as an antigen-presenting cells. Thus, the innate immune response to infection hastens the transport of antigens to the local lymphoid tissue and enables those cells that have taken up antigen to present it effectively to the naive T cells that migrate through this tissue. Have a great day.